Welcome to the second part of Module 8 on genomic selection, where we were discussing other considerations. We ended the first part of this module by talking about ways to subset your data, throwing out marker data from your training population and throwing out lines from your data population to increase your accuracy by reducing the noise in your data set and also by reducing redundancy. One thing we can do is throw out lines in our data in our training population that have a lot of genotype by environment interaction associated with them. And this I'm just showing you some of the results from doing that in our training population. And let's just look at one trait, Fusarium head blight resistance in wheat. We started out with a training population of 470 lines, and that gave us an entry mean heritability of 0.45. We then went through and figured out the genotype by environment interaction variance associated with each of those lines. Throughout the 70 that had the most G by E associated with them, ended up with 400 lines, and now we have an entry mean heritability of 0.64 amongst those remaining lines. And that should result in higher GS accuracy because we've thrown out a lot of noise from our data set, increasing the proportion of the data that's associated with pattern, and therefore it has predictive ability. And you can see we do this with yield, and we get the same results. And this can increase our accuracy of genomic selection. So here's our accuracy that we obtained when we used all lines, data from all lines in the training population to build a GS model. And here is accuracy we got within environments, shown in these data points up in this part of the graph. And here's the accuracy we got between environments. And this one is particularly important. So using all data, our GS accuracy between environments was actually quite poor. In fact, it was negative. That's worse than poor, it's terrible. But if we throw out the unstable lines, using only the stable lines to build our GS model, we now have accuracy of an average about 0.3 between environments. Within environments, the average GS accuracy using all lines in the training population to build the model was 0.72. And when we threw out the unstable lines and used only the stable ones to build our models, the average accuracy was 0.8. So we were able to get some increases by using only the stable lines. Now let's talk about genomic selection and marker-assisted selection. These, again, are not exclusive approaches to doing molecular plant breeding. They are actually quite complementary to each other. Marker-assisted selection can be used to quickly fix genes with large effects in your population, and then genomic selection can be used to select the best values for the rest of the genome. You can actually get separate marker data from for marker-assisted selection and GS-derived prediction models. For example, you might want to use genotyping by sequencing to get whole genome coverage to do GS, but you might want to use CASP assays to get marker data to do marker-assisted selection for a few key genes. And here's just an example of data again out of my program where we have different F2 individuals that we are now going to predict their value. And here is their predicted value for yield in one of my locations. So here's their values here, and if they're high values are the ones we want. Low values, which are shown down here, are the ones we do not want. And what I'm showing in these four columns is the presence or absence of favorable alleles for yield that we have established. If the cell is green, that means for that marker we have the presence of a favorable allele for increasing yield. And if it's the cell is unfilled, it means it has either missing data or it is has the allele for decreasing yield. So what we can do here is we certainly want to pick the individuals towards the top here that have the highest predicted values for yield, but also we can want to make sure we pick the individuals that have the alleles associated with high yield at these QTLs. Now if I cross between these kind of lines that are shown in these boxes, what I'll be doing is actually fixing the QTLs for increased yield, but also selecting for high estimated breeding values for the rest of the genome. And that, of course, is our goal in plant breeding. Fix the favorable alleles, increase the value of the genome. Genomic selection and genetic diversity. So you're going to go through, you can start your training population, start crossing, getting individuals, and going through cycles of genomic selection. What's going to happen to your genetic diversity within these populations are going through rapid cycles of selection. And most studies show that diversity within a population will decrease as you go through cycles of genomic selection. And perhaps the change is even more rapid than what you might get if you're doing phenotypic selection. So, and part of that is expected, and part of it is not good. But you think about this, within a population, your goal is to fix the favorable alleles. So in that sense, you actually want to lose diversity. In other words, you want to lose the bad alleles. But also, when you go through some of these cycles, and you go through them rapidly, and you have somewhat small populations, you may be losing diversity just through random genetic drift. And of course, that is unfavorable. You want to fix the favorable alleles, but you also run the risk of losing favorable alleles simply to drift alone. One thing about this is, as you're going through these cycles of selection, you have marker data, and these data provide you a way to monitor the loss of diversity, monitor the fixation of alleles, and it basically changes in your whole genome. So there's some nice ways to monitor what's happening there. Here's some data out of my program again. We're looking at simply the simple matching coefficient of the training populations in cycle 1 and cycle 2 of F2s I've gone through genomic selection. A simple matching coefficient is really just a proportion of alleles that are common between any two lines. So a low simple matching coefficient indicates two lines that don't have many genes in common, and the high value indicates that the lines have more alleles in common between them. So we started out our mean simple matching coefficient in our training population was about 0.64, and after 2 cycles of genomic selection, it is now up to about 0.68. So it doesn't indicate our lines are becoming more related and we're losing diversity. Interestingly enough, as we look at our what is the minimum simple matching coefficient, in the training population it was 0.5. In other words, basically the most diverse any two lines in my training population is 50% of alleles shared and 50% different. And after 2 cycles of genomic selection, I can still find pairs of lines that have a very low simple matching coefficient equal to that we found in the training population. Interestingly, the variation in our simple matching coefficient continues to go up, and I'm not sure exactly what that means. We can also look at how our individuals in our cycles of genomic selection, how do they fill the genetic space that's also occupied by our training population. And what we're showing here are our training population lines are shown in blue. No, I better go back. Here are our training population lines are shown by numbers, which indicate their cluster. And the cycle 1 F2s are shown by asterisks. And basically the whole gist of this slide is that our lines are coming out of cycles of genomic selection are still remaining about the same, occupying about the same genetic space as our training population did itself. So that's good. We want to be sampling the same genetic space. Another aspect of genomic selection is what is the end product of cycles of genomic selection. It's not necessarily a single new variety. And it's not necessarily identification of certain markers with certain traits. In other words, QTLs being identified. The end product is really an improved population. After each cycle, there's a selected set of F2, F3, F4 derived families. All right, and there's a variation amongst them. They are not all genetically the same. It's a heterogeneous population. And this heterogeneity can be, of course, between the families derived from genomic selection, but also within them. If you're selecting F2s and you're getting F2 derived families, there's still a lot of heterogeneity within those families. And this really provides a segregated, you might look at this and say that's a big disadvantage, but there's also opportunities here. This is a segregating resource that you have generated, and you can then share it with partners and colleagues. It actually could be quite useful maybe in participatory plant breeding, where no further molecular technology is required to benefit from it. You've created this improved population, you hand it off to someone else who may just do traditional plant breeding with it. So, and here's just an example of that. You start with F1s, you get your F2 population, you can do genomic selection in it. These are the best individuals. Okay, you cross with them, but you also self them. You get this family, and from there you can drive other families. This resource now can all be shared with your colleagues and distribute to anyone else who may want to do selection in it. That is the end of this part of the module, and it'll be continued in the third and final module section of the module.